Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore a truly fascinating topic, pigmentation in the Ephestia moth. We often think that an organism's traits are determined entirely by the DNA it inherits from both parents at the moment of conception. But what if I told you that sometimes the mother's genetic makeup alone can completely dictate how her offspring look when they are born, regardless of the offspring's own genes? This phenomenon is called the maternal effect, and it is one of the most beautiful departures from standard Mendelian inheritance. Let us dive in and see how this works. To understand our topic, let us start with a simple analogy. Imagine you are going on a long, challenging hiking trip, and your mother packs a large backpack full of highly nutritious food for you. Even if you do not know how to cook or forage for yourself, you will survive and thrive during the early days of your trip simply because of the supplies she provided. In the biological world, a very similar and fascinating phenomenon occurs during the early development of certain organisms. Sometimes a mother packs essential proteins, messenger RNAs, or chemical precursors directly into the egg cell before it is even fertilized by the father. This biological packed lunch kickstarts early embryonic development. Now that we have our analogy, let us define what this means scientifically. This phenomenon is known formally as the maternal effect. In a true maternal effect, the physical appearance, or phenotype, of an offspring is determined strictly by the nuclear genotype of its mother. It completely ignores the offspring's own newly formed genotype, and it completely ignores the father's genetic contribution. Because the mother loaded the egg with specific biochemicals based on her own DNA, the baby organism will display traits based entirely on what she provided. This is a beautiful departure from standard Mendelian inheritance, where we usually expect both parents' genes to immediately influence the offspring. So, how does this look in a real organism? The pigmentation in the Mediterranean flower moth, scientifically known as Ephestia quainiella, is a textbook example of this genetic phenomenon. In these moths, creating brown pigment in the eyes and the larval skin requires a specific chemical precursor called kynurenine. Without kynurenine, there is no brown pigment. During egg production, a mother moth who is capable of making kynurenine will actively pump this chemical into the cytoplasm of all her developing eggs. She's essentially packing that biological lunch we talked about, filling the eggs with the precursor needed for pigmentation before fertilization even occurs. Before we look at the crosses, we need to understand the genetics behind kynurenine production. The ability to manufacture this chemical is controlled by specific alleles. The dominant allele, capital A, contains the functional genetic instructions to manufacture kynurenine. On the other hand, the recessive allele, lowercase a, is defective and cannot produce it. Therefore, when a mother moth possesses at least one dominant A allele, her body successfully produces kynurenine. She will generously deposit this kynurenine into the cytoplasm of every single egg she produces, regardless of which specific allele that individual egg will eventually receive during meiosis. Now that we understand the alleles, let us look at the mathematical expectation for a standard test cross. This formula represents the expected genotypic ratio of the offspring. F1 represents the first filial generation, which simply means the direct offspring resulting from a parental cross. The term capital A, lowercase a represents the heterozygous genotype, meaning the offspring inherited one functional dominant allele and one defective recessive allele. The term lowercase a represents the homozygous recessive genotype, meaning the offspring inherited two defective alleles and cannot produce its own pigment. The numerical value 0.5 indicates that exactly half the offspring will have one genotype and half will have the other. While this formula perfectly describes the DNA inherited, the maternal effect will alter the physical outcome. Let us apply this formula to a concrete example. Suppose we have a female Ephestia moth with a heterozygous genotype, capital A, lowercase a, and she is mated with a male moth that has a homozygous recessive genotype, lowercase a, lowercase a. We need to determine the physical appearance of their larval offspring immediately after hatching. First, we look exclusively at the mother. Because she is heterozygous, she produces kynurenine and packs it into all her eggs. Next, we calculate the offspring genotypes, which gives us a 1-to-1 mix of heterozygous and homozygous recessive larvae. However, because the mother provided kynurenine to all eggs, every single larva that hatches will initially have pigmented skin and dark eyes. The maternal cytoplasm overrides their own genetics. This surprising result leads us to an important question. What happens if we swap the parents? To find out, we use this formula for a reciprocal cross. Here, PF represents the female parent moth, who is now homozygous recessive, meaning she is unpigmented. PM represents the male parent moth, who is now heterozygous and pigmented. The arrow represents the biological process of reproduction and fertilization leading to the offspring generation. Notice that the genetic outcome for the F1 generation is exactly the same as our previous example, a 50-50 mix of heterozygous and homozygous recessive offspring. But because the mother's genotype has changed, the physical traits of the offspring will be drastically different. Let us solve this reciprocal cross step by step. We are given a homozygous recessive mother and a heterozygous father, and we need to find the offspring's early phenotype. Our first and most important step is to examine the mother's genotype. Because she is lowercase a, she completely lacks the functional allele required to synthesize kynurenine. Therefore, she leaves her eggs biochemically empty. The father's ability to make kynurenine is completely irrelevant right now because the sperm only contributes tightly packed DNA, not the bulky cytoplasm needed to store chemicals. Even though the offspring genotypes are the exact same 50-50 mix as before, every single larva will hatch completely unpigmented and pale because they started with absolutely zero kynurenine from the mother. Now that we have calculated both examples, let us use them to formally prove the maternal effect. In this first part of our proof, we compare the immediate results of the two crosses. In cross 1, the offspring were a 50-50 mix of genotypes, but 100% of the larvae hatched with pigment. In cross 2, the offspring had the exact same 50-50 mix of genotypes, yet 100% of the larvae hatched without pigment. Mathematically and genetically, the offspring pools are identical. The only variable that changed was which parent provided the egg. This proves that the early physical traits of the organism are entirely dictated by the mother's genetic ability to provision the egg prior to fertilization. Continuing our proof, we must look at what happens as these larvae grow into adults. In cross 1, the homozygous recessive larvae initially had pigment, but as they grow, they use up the finite supply of maternal kynurenine. Because they lack the functional A allele, they cannot make more, and they eventually develop into unpigmented adults. Conversely, in cross 2, the heterozygous larvae hatched pale, but as they grow, they eventually activate their own functional genes. begin producing their own kynurenine, and slowly develop pigmentation. This final result proves a crucial point. The maternal effect controls the early embryonic stages, but it is temporary. Eventually, the offspring's own DNA wakes up and takes over their physical development. Let us visualize everything we have just proven with this diagram of the reciprocal crosses. On the left, we see cross 1. Notice the pigmented mother at the top. The arrows show her depositing kynurenine into the eggs, resulting in a cluster of fully pigmented brown larvae at the bottom, despite their mixed genetics. Now look at the right side, cross 2. Here, the mother is unpigmented. The diagram clearly shows that her eggs lack kynurenine, leading directly to a cluster of pale, unpigmented larvae. By comparing the two sides, you can visually confirm that the father's appearance has no impact on the newly hatched larvae. The early phenotype is a direct, undeniable reflection of the mother's genetic makeup. Before we conclude, we must understand why this concept is so absolutely crucial in genetics. The maternal effect shatters the simplistic illusion that an organism's traits are instantly and solely dictated by its own DNA from the exact moment of conception. Instead, it highlights the profound importance of the maternal environment, specifically the oocyte cytoplasm, in kickstarting early embryonic development. If geneticists did not understand this, they would be completely baffled by breeding experiments where offspring phenotypes wildly contradict expected Mendelian ratios. Furthermore, this concept extends far beyond moths. It provides critical insights into certain human genetic conditions and developmental anomalies that are heavily influenced by the mother's metabolic state prior to fertilization. We have covered a lot of ground today, so let us summarize our key takeaways. First, the maternal effect is a powerful phenomenon where early physical traits are dictated entirely by the mother's genetic ability to provision the egg. Second, in the Ephestia moth, mothers with the dominant A allele pack their eggs with the pigment precursor kynurenine, making all their newly hatched larvae brown. Third, by using reciprocal crosses, we proved that identical offspring genotypes can result in drastically different early phenotypes, depending solely on the mother. Finally, remember that this effect is temporary. As the maternal supplies run out, the offspring's own genes take over. You now have a solid understanding of the maternal effect. Great job!